Usually when we think about the biology of healing, we imagine it as this purely benevolent force.
Right. Sort of like a tiny microscopic team of paramedics.
Exactly. I mean, you get a cut on your skin or a scrape on your knee, and this incredible, highly coordinated molecular machinery just kicks in. It stops the bleeding, it repairs the tissue, builds new scaffolding, and essentially, makes you whole again.
It's a really comforting way to look at our biology.
Mhm.
We naturally want to categorize cellular functions as strictly good or strictly bad. Like healing is good, disease is bad.
The lines seem very clear.
They do, yeah. But then you step into the actual mechanics of tissue remodeling, and suddenly that neat categorization completely falls apart.
It really does. You realize we are looking at a biological landscape that is incredibly paradoxical. Because what if I told you that the exact same receptor on your cells that helps your tissues heal, the one that gives your skin its structural integrity, is the exact same molecular machinery that cancer stem cells use to spread throughout the body?
It forces you to rethink how the body works. I mean, the very system that keeps you put together, that patches you up, is the identical mechanism that can tear everything apart if it gets hijacked.
And in today's deep dive, we're looking at a huge stack of sources. We've got everything from high-resolution structural protein databases to clinical pathology papers, all to figure out how this paradox actually functions.
Yeah, we are zeroing in on one specific shape-shifting protein on the surface of our cells. It's called CD44.
And the stakes here are massive for you, the listener, because it's not just hijacked by cancer. We're learning that everyday environmental pollutants can trigger this exact same receptor to cause severe lung fibrosis.
Which makes CD44 arguably one of the most important yet, widely misunderstood glycoproteins in the human body. To really grasp how it controls our fate, we have to understand that it operates basically like a cellular communications hub.
Right, an antenna.
Exactly. It sits on the outer boundary of your cells, taking in physical and chemical messages from the environment. And it dictates whether a cell behaves itself and stays put, or if it goes completely rogue, starts dividing rapidly and migrating where it absolutely shouldn't.
I want to understand how a single protein can wield that much power. And the sources point out that to do that, we can't just talk about it in the abstract. We have to really zoom in.
We have to zoom way in.
Right. We're talking about looking at data from the protein data bank. Specifically, there's a file called PDB-ID1UUH, which freezes this protein so we can look at it down to a resolution of 2.20 angstroms, which is, it's hard to even conceptualize.
It's incredibly small.
Wow.
We're looking at the spaces between individual atoms at that point.
So what do we actually see when we get that close?
Well, when you zoom into the atomic level, you see that CD44 is an 80 to 90 kilodalton type I-transmembrane glycoprotein.
Okay, wait, I'm going to need a translation on that. That sounds like a piece of industrial hardware.
Fair enough. Let's break the physical reality of it down. Kilodalton is just a measure of weight, so it's a relatively hefty molecule. And transmembrane just means it spans across the cell's outer boundary.
Like a bridge.
More like a buoy floating in the ocean. Part of it is up in the air, part of it goes right through the surface of the water, and part of it hangs down into the ocean below. CD44 has an extracellular domain that sticks out into the environment, a middle section anchored through the cell membrane, and a cytoplasmic tail that hangs down inside the cell.
Got it. And the glycoprotein part, does that mean it's coated in sugars?
Yes, proteins often have complex sugar molecules attached to them to help with stability and function. But structurally, the most fascinating part is that outside piece, the extracellular domain.
The part sticking out into the environment?
Right. At 2.20 angstroms, you can see it has this highly structured globular region. And it doesn't just flop around randomly. It is held in a very specific, rigid 3D shape by something called a tridisulfide bond ladder.
Wait, a ladder made of sulfur bonds?
Essentially, yes. They act like biological rebar. These chemical cross-links ensure the protein holds a very specific shape. And protruding from that rigid structure is a lobular extension.
So it has this rigid, highly specific shape, and the whole point of that shape is to catch things floating by. It acts like an antenna, or maybe more accurately, a catcher's mitt.
A catcher's mitt is a perfect way to visualize it.
And it's primarily looking to catch one specific molecule, right? Hyaluronin, or HA, which, from what I gather from the sources, is this giant sugar polymer. It acts like biological mortar holding our cellular bricks together.
That's the primary ligand, yes.
Yeah.
HA is the key that fits into the CD44 lock. But we shouldn't think of it as just two puzzle pieces snapping together randomly. The high-resolution data shows a highly specific HA binding domain.
Meaning it has a specific grip.
Exactly. It relies on a very precise arrangement of amino acids inside that catcher's mitt, specifically lysine 38, arginine 41, tyrosine 42, and arginine 78.
I want to picture the physics of this. Why those specific amino acids? Do they act like the fingers of the glove?
You can think of them as the fingers, sure, but it's really about magnetism and charge. Amino acids like lysine and arginine carry a positive electrical charge at normal physiological conditions.
I see where this is going.
Right. Hyaluronan, on the other hand, is a polyanion. It is heavily negatively charged. So the receptor creates this perfectly shaped pocket lined with positive charges, just waiting for this negatively charged sugar polymer to float by.
And when they align?
Opposites attract. The mitt closes around it, and the physical signal is triggered down into the cell.
Okay, so the outside of the cell has this rigid, positively charged mitt, and it catches this negatively charged HA molecule. But here is where I get really confused by the sources. The HA molecule isn't always the same size.
No, it varies wildly.
You have high molecular weight HA, which is this massive long chain, and you have low molecular weight HA, which are basically chopped up tiny fragments. And supposedly the cell reacts completely differently depending on which one the mitt catches.
It's a night and day difference.
But it's the exact same chemical. How does a cell possibly know if it's catching a big piece or a small piece of the exact same sugar?
The cell knows because the physical tension on the membrane changes completely. It basically comes down to the mechanics of what we call multivalent binding.
Multivalent binding. Okay, explain that.
Imagine that high molecular weight HA is like a massive heavy parachute draped over a crowd of people.
With the people being the CD44 receptors.
Yes. Because that parachute is so huge, it touches many different people or receptors at the exact same time. It physically links them together, clustering them into a stable, quiet lattice on the surface of the cell.
Oh, wow. So they're all tied together.
Right. When CD44 receptors are clustered like this by a giant HA molecule, they send a protective calming signal into the cell. It acts as an anti-angiogenic and anti-inflammatory signal.
Meaning it stops abnormal blood vessel growth.
Exactly. It tells the tissue to relax.
So the massive HA chain essentially gives the receptors a giant, calming, weighted blanket. They're anchored in place. But what happens when there's tissue damage and that giant chain gets chopped into tiny pieces?
Well, the blanket is gone. Now imagine cutting that massive parachute into tiny individual squares. Each little square can only bind to a single receptor.
So there is no clustering anymore.
None at all. The receptors are freed up, they become highly active, and they start moving independently. The physical tug on the receptor is completely different, and that change in mechanical tension sends a fundamentally different signal inside the cell.
That is wild. The receptor is physically feeling the size of the molecule based on how much it gets dragged around. And that new signal from the chopped up HA, the low molecular weight stuff, that's the danger signal, right?
Dramatically so. Instead of keeping the cell calm, those smaller HA fragments promote cell motility, meaning the cells drop their anchors and start migrating.
They just take off.
Yeah, it promotes the building of new blood vessels, and it flips the cell into a state of aggressive tissue remodeling. In fact, the sources note that CD44 exists in 3 states: inactive, inducible active, and constitutively active.
With constitutively active, meaning it is permanently jammed in the on position, right? Which is what you see in tumor cells.
Exactly. They hijack the receptor so it's constantly screaming at the cell to grow and move, regardless of what the HA outside is doing.
And the complexity doesn't stop there, because if the outside environment is constantly changing, the cell needs to adapt its antenna. It isn't just the HA ligand that changes size. The CD44 receptor itself is a shapeshifter.
It is incredibly adaptable.
I was trying to wrap my head around this part of the research. The genetic blueprint for this receptor, the CD44 gene, has 20 different exons, which are the coding sections of the gene. 10 of them are standard, meaning every CD44 receptor gets them.
Yes, that's the baseline.
But the other 10 are variant exons, which are subject to alternative splicing.
When we talk about alternative splicing, we are talking about the cell's ability to edit its own hardware on the fly. Depending on the tissue type or, if a cancer is rapidly developing, the cell will splice in different combinations of those variant exons.
So it literally rebuilds the receptor.
Yeah, it physically changes the length of the stalk holding the catcher's mitt.
It reminds me of buying a modular smartphone. You start with the base model, the 10 standard exons. But depending on what you need to survive today, you can snap on a specialized macro camera lens or maybe a heavy-duty battery pack just by splicing in those variant exons. You are literally customizing the hardware to fit the environment.
The modular smartphone is an excellent way to visualize it. And the cell does this not just through genetics, but after the protein is already built and sitting on the membrane.
Oh, really?
Yeah, it undergoes post-translational modifications. Two critical ones are N-glycosylation and polymethyylation.
Okay, N-glycosylation. I know we defined glycoprotein earlier as having sugars attached, so I assume this is the cell physically attaching more sugars to the receptor.
Yes. The cell attaches complex, branching sugar chains right near the catcher's mitt. And the structural data shows this acts like a physical tuning dial.
A tuning dial? How so?
Depending on how heavily glycosylated the receptor is, those bulky sugar chains can physically block HA from entering the mitt, or they can alter the shape just enough to enhance the grip.
That's brilliant. The cell can tune the affinity of the receptor without having to build a completely new one. But what about polymethyylation? That sounds like we were dealing with fats now.
We are. Polymethyylation is a lipid modification. The cell attaches a fatty acid chain to the receptor right where it passes through the cell membrane.
Okay, why would it do that?
To understand why this matters, you have to picture the cell membrane not as a uniform wall, but as a fluid ocean of lipids. Within that ocean, there are specific stiff islands called lipid rafts.
Lipid rafts?
Yeah, these rafts are packed with cholesterol, and they are essentially the VIP sections of the cell surface where all the heavy-hitting signaling molecules congregate.
And because fat likes to stick with fat, attaching that fatty palmitate chain to the receptor drags it into the lipid raft.
Precisely. If CD44 isn't polymethylated, it floats aimlessly in the fluid part of the membrane. But once that fatty anchor is attached, it gets dragged into the lipid raft right next to the most powerful signaling proteins in the cell.
So without that modification, it can't effectively pass its messages down the line.
Exactly. It needs to be in the VIP section to be heard.
Which brings us perfectly to the inside of the cell. We've talked about the outside mitt catching the molecule. We've talked about the stalk being customized and dragged into the VIP section. But what is actually happening on the inside?
That's where the real action is.
Because the part of the receptor that hangs inside the cell, the cytoplasmic domain, is remarkably short. I kept wondering, how can such a tiny internal tail possibly orchestrate massive cellular changes?
It is a profound structural question. But despite being tiny, that cytoplasmic tail physically anchors the entire receptor to the cell's internal skeleton, the actin cytoskeleton.
So it's plugging directly into the cell's structural bone.
Yes, it uses specialized linker molecules called ankyrin and ERM proteins. That's ezrin, radixin, and mosin. This creates a direct physical wire from the extracellular environment straight through the membrane all the way down into the structural scaffolding of the cell.
So if the outside gets tugged, the inside skeleton immediately feels it.
Exactly.
But it's not just structural tension, right? The literature describes this tail as the central hub for massive chemical signaling cascades, pathways that tell the cell to survive and block apoptosis, which, for our listeners, is programmed cell death.
Right, the PI3K and AKT pathways.
Yeah, and pathways that drive intense, relentless cellular multiplication, like the SRC and MAPK pathways. But how does one tiny tail trigger all of that simultaneously? The math just doesn't seem to work.
The math works because the tail doesn't actually do the chemical heavy lifting itself. It acts more as an integrator.
An integrator.
Yeah. Think back to those lipid rafts, the VIP sections. When CD44 catches chopped up AJ and it's gathered in that lipid raft, the tiny internal tail acts like a shepherd, or maybe a party promoter.
A party promoter.
It physically interacts with and drags massive co-receptors into the same physical space. We're talking about heavy hitters like c-Met, EGFR, and TGF-beta.
Oh, I see. So it's not firing the signals itself. It's pulling the proteins that do fire the signals into a tight little huddle so they can work together.
Exactly. It forces these growth proteins, the kind of proteins that tell a cell to multiply relentlessly, into one tight cluster. By bringing the right players to the table at the exact same time, they fire off their signals in tandem.
Causing a massive coordinated response.
Right. It amplifies the signal tremendously.
So we have this incredibly elegant, highly tuned system, an atomic-level mitt catching signals, adapting its shape, dragging proteins together, and wiring the outside world to the inside skeleton. But what happens when the system goes rogue?
That is the dark side of tissue remodeling we promised to look at.
Let's look at fibrosis first. Fibrosis is, at its core, a wound-healing response that just refuses to shut off.
Right. If you have severe lung fibrosis, your tissue is replacing itself with rigid scar tissue, making it impossible to breathe. And CD44 is central to this. When the receptor catches low molecular weight HA, those chopped up danger signals, it stimulates fibroblasts to pump out massive amounts of extracellular matrix.
It puts the cells into overdrive, laying down biological mortar until the tissue is just a solid block of scar.
And clinical data indicates that severe lung fibrosis literally requires this specific HA-CD44 pathway to be active. If CD44 doesn't misfire, the runaway scarring doesn't happen.
And it's the exact same story with cancer. The sources point out that CD44 isn't just incidentally involved in tumors, it is a major marker for cancer stem cells.
Yes, because cancer hijacks a deeply insidious biological process called EMT, the epithelial mesenchymal transition.
Okay, we definitely need to translate EMT. What does it actually mean for a cell to undergo that transition?
Think of epithelial cells as the bricks in a wall. They line your organs, they're stationary, and they form tight barriers. Mesenchymal cells, on the other hand, are highly mobile, almost like an amoeba.
Okay.
EMT is the process where a stationary brick drops its anchors, changes its entire shape, and becomes a highly mobile, invasive cell.
Literally a brick turning into a motorized vehicle.
Yes, it's a natural process during embryonic development, but cancers hijack EMT to metastasize and spread through the body. And that process is driven heavily by CD44.
So the cancer cell uses the receptor to escape.
Exactly. When the cancer cell splices its CD44 into those variant forms we discussed, the internal tail fires off survival signals so the cell doesn't die while traveling in the bloodstream. And at the same time, it physically reorganizes the cell skeleton to give it the motor it needs to move.
That is terrifyingly efficient. And what makes this feel so urgent is that it's not just internal genetic glitches triggering this. The research highlights that our modern environment is now playing a massive role in hijacking this exact receptor.
We are seeing that environmental pollutants, heavy metals, and specifically microplastics are now interacting with this system.
Microplastics.
Yeah. When foreign materials like microplastics enter your tissues, say you inhale them deep into your lungs, they cause microscopic physical damage and chronic inflammation.
And that inflammation and physical shearing chops up the giant, calming hyaluronan blankets into those dangerous, low molecular weight fragments.
Which then bind to your CD44 receptors. The cell gets the signal that there's a massive wound that needs urgent repairing. It triggers the fibroblasts to start laying down scar tissue.
But because the microplastics don't go away, the wound never actually heals.
Exactly. The CD44 pathway stays locked on, and you develop progressive fibrosis.
It's a chilling reality. The idea that inhaling everyday pollutants is artificially pressing the exact same CD44 button that our body naturally uses for repair, but tricking the lungs into forming deadly scar tissue by mimicking a wound that never closes.
It perfectly illustrates why mapping this receptor at a 2.20 angstrom resolution isn't just an academic exercise. This protein sits at the exact intersection of healthy repair and fatal disease. Understanding its physical shape is the only way we can learn to control it.
We have covered a tremendous amount of ground today. I mean, we started with a biological paradox, a single receptor that heals and harms. We zoomed down to the atomic level to see the positively charged amino acids forming a catcher's mitt.
Right, catching the HA.
And we saw how a massive sugar polymer acts like a calming weighted blanket, while chopped up fragments trigger a cellular panic.
We also explore the structural adaptability, how the cell splices new parts like a modular phone, tunes its antenna with sugars, and uses fatty anchors to drag the receptor into the VIP lipid rafts.
And we saw how a tiny internal tail acts as a shepherd for massive growth signals, ultimately driving the runaway trains of fibrosis and cancer metastasis, especially when triggered by environmental damage.
It really is a masterclass in cellular complexity.
So, knowing all of this, I want to leave you with a final provocative thought to mull over. If we have mapped this protein so perfectly, if we know the exact position of lysine 38 and arginine 41 in that catcher's mitt, and we know that tiny, chopped up fragments of HA trigger the disease pathways while the massive ones protect us, could we design a perfectly shaped synthetic molecule that plugs up that receptor?
You're talking about a molecular cork.
Exactly. A microscopic cork that fits so perfectly into the CD44 lock that it prevents environmental pollutants or chopped up HA from binding and triggering cancer or fibrosis, but engineered so specifically that it leaves the healthy, heavy blanket HA pathways completely untouched. Could we literally engineer our way out of the paradox?
That is the ultimate frontier of structural biology.
Yeah.
I mean, if we can see the lock at an atomic level, it is entirely possible we can design a new key.
And suddenly, all that dense data from the protein databank isn't just abstract science anymore, the muddy waters clear up, the paradox isn't a dead end, it's a blueprint for a cure. Until next time on the deep dive, keep asking questions.